Because there is a vast microbial diversity, scientists have classified one organism together with the other microorganism having similar property with the other. The science of classification, especially the classification of living forms, is called taxonomy, from the Greek for orderly arrangement. The objective of taxonomy is to classify living organisms, that is, to establish the relationship between one group of organisms and another to the different or to differentiate them. There may be as many as 100 million different living organisms, but fewer than 10% have been discovered, much less classified and identified. Taxonomy also provides a common reference for identifying organisms already classified. For example, when a bacterium suspected of causing a specific disease is isolated from a patient, characteristics of that isolate are matched to list of characteristics of previously classified bacteria to identify the isolate. Finally, taxonomy is a basic and necessary tool for scientists, providing a universal language of communication. And since we are talking about microorganisms, scientists have come up with the classification, namely prokaryotes, eukaryotes, and those that is unique from the previous mentioned group, which are atypical cells. So in this topic, we will unravel each of them. First is the prokaryotes as gram-negative. The first group belonging to this domain is Enterobacteriaceae. Enterobacteriaceae is a facultative anaerobes. Facultative anaerobes means that they have the faculty to live with oxygen. Let's review first on some concept about oxidation. When we say oxidation, it can kill. Even in our cells, if it was oxidized, what will happen? It will become dry and no life and will result in aging. That's why we take antioxidant to have a glowing skin, right? Bacteria also are like that. But because there are enzymes that help bacteria to live with oxygen, they can now live with the presence of oxygen by catalyzes the breaking down of different substances making oxygen. These enzymes are catalase, or let's abbreviate it as CAT, peroxidase, or let's abbreviate it as PER, and superoxidase, or let's abbreviate it as SUPER. Depending on the following enzymes, bacteria can tolerate oxygen. Like for example, we have the obligate aerobes. Obligate aerobes cannot live without oxygen. Review from our previous discussion, obligate means that they can live on one specific environment only. Aerobes means oxygen or there is a presence of air. So this means that obligate aerobes can only live with the presence of oxygen. But why they can tolerate oxygen or why they can live with the presence of oxygen? This is because of these three enzymes, which is the cat per super or the catalase, peroxidase, and superoxidase. So because of these three enzymes, the oxygen in their environment can break down oxygen and help them live with such environment. Again, they have these three enzyme, which is the cat, per, and super, to live with the presence of oxygen. Another example is the facultative anaerobes, which is the Enterobacteriaceae in our first gram-negative example. Facultative anaerobes are not actually anaerobes. So means they are not actually anaerobe but rather aerobes. Because they have the super and cat enzyme that gives them the faculty to tolerate conditions with or without the oxygen. Facultative, from our previous discussion, means they can live with more than one kind of environment. So in our case, facultative anaerobes can live with the presence of oxygen and without the presence of oxygen. And again, why they can live in the presence of oxygen, it is because they have these two enzymes, which is the superoxidase and catalase. Another example is bacteria, which are considered as microaerophilic. Microaerophilic are bacteria that has superoxidase enzyme only. Because it only has one enzyme, it can only tolerate small amount of oxygen. That's why it's called micro, means small or little, aero means oxygen, and philic, which is loving. So it means that they are loving with only small or little amount of oxygen. So the number of enzyme for microaerophilic then is only one. That's why they can only tolerate little amount of oxygen because they only have one enzyme to break down oxygen. Lastly, we also have bacteria which cannot live with the presence of oxygen. They are obligate anaerobes. So obligate anaerobe has no enzyme to help them live with a condition that has oxygen. Because of this, they can die with the presence of oxygen. Going back with Enterobacteriaceae. Since we know that Enterobacteriaceae is facultative anaerobes, they can either live with the presence of oxygen, they can also live without the presence of oxygen. Enterobacteriaceae, aside from facultative anaerobes, are also considered as rod-shaped bacteria. So it's look like this in the picture. They are also considered as non-sporing. Since they are considered as non-sporing, we know that there is no spores for the DNA and RNA. Enterobacteriaceae are also considered as motile. When Enterobacteriaceae are motile, they have this peritracheous flagella that helping them to move, except for Shigella and Klebsiella that doesn't have the flagella. Since Shigella and Klebsiella are bacteria that doesn't have flagella, they are non-motile or they cannot move freely. Enterobacteriaceae also considered fermenting glucose with acid production, in which this acid that they are producing is the lactic acid. That is why the agar that can be used is McConkey's agar, and we discussed this already last meeting why McConkey's agar can be used for those bacteria that can. Okay, the red text here are the descriptions that was expected because they belong to this group, which is Enterobacteriaceae. So we will focus mainly on the black text color. Since these bacteria can be found on gastrointestinal, we are expecting that the disease it can give also are GI-related diseases. Since these bacteria can be found on gastrointestinal, we are expecting that the disease it can give also are GI-related diseases. But you might wondering if it is primarily affecting the GI, why does it has symptoms that is far from the outside of the intestine? So basically, it is because of the toxin it produces. The toxins they are producing are enterotoxin and endotoxin. Enterotoxin is a toxin produced in or affecting the intestine. This toxin will be secreted by the microbe like the E. coli towards its environment, which is specifically once it reaches the intestine. This is the reason why the affected host has the symptoms like gastroenteritis, or if you see itis on the disease, you know it's inflammation. So gastroenteritis is an inflammation in the stomach and intestine caused by the bacteria E. coli. Symptoms of gastroenteritis are watery diarrhea, stomach pain, and vomiting. Also, sepsis can be observed when the bacteria was triggered then it will give severe diarrheal symptoms. Or if it on the lower part of the intestine, UTI may also be observed. Endotoxin, on the other hand, is a toxin present inside a bacterial cell wall, and it is released when it is disintegrates. So this means that this toxin will affect its host once it will be broken down, because this toxin is present on the cell membrane of the bacteria. We know that our body also fights foreign microbes in the body, right? So tendency would be, if our microflora or the good bacteria innate in our body detected that there is a presence of E. coli, it tried to kill E. coli by breaking its cell membrane, and if that happens, endotoxin will be secreted. The problem with this is that since depending where E. coli is located, once it releases the toxins in that particular area, it causes disease. Example, when an 8 to 28 days old baby contacted E. coli, it may cause neonatal meningitis. Neonatal meningitis will happen to the baby once E. coli reaches the meninges, which located in the brain, and then was disintegrate in that area. Or if the patient was admitted to the hospital and after 48 hours or more, pneumonia was detected, then this means that E. coli was disintegrated in the lungs of the patient. This type of pneumonia is also known as HAP, or hospital-acquired pneumonia. This means that this pneumonia was only acquired during the admission to the hospital and not before the patient was admitted. E. coli was the most common causative agent also of hospital-acquired pneumonia. Treatment for E. coli are cotrimoxazole or quinolones. E. coli has several classifications. Each strain will also has a specific severity or target once it is on the body. EPEC, or the enteropathogenic E. coli, will give diarrhea in infants and children. For me not to forget that, I always associate the EPEC or this strain of E. coli having the P letter, that means pedia or pediatric. So pedia or infant and children. So since this is next is the ETEC, or the enterotoxigenic E. coli, or also known as causing travelers diarrhea, or also known as Montezuma's revenge. So why it is called as Montezuma's revenge? Montezuma II was emperor of Mexico from 1502 to 1520 and was in power when the Spanish began their conquest of the Aztec Empire. So every time there is a white that got diarrhea that go to the Mexico, they said that they are suffering from Montezuma's revenge. The sickness also known as the traveler's diarrhea. This is usually caused by drinking the local water or eating food that visitors aren't accustomed to. It is a bacterial illness, always uncomfortable and occasionally serious. It can be either a mild or explosive illness. Most cases are caused by the E. coli bacterium. The revenge element of the phrase alludes to the supposed hostile attitude of countries that were previously colonized by stronger countries, which is the Spaniards, which are now in the small but effective way getting their own back. So, to not forget that ETEC caused travelers diarrhea, I always associate this that it has the T letter on its name, so I associated with T letter as travelers diarrhea. Aside from the two strain, we also have the EAC or EIEC, or the enteroinvasive E. coli, that is similar to shigellosis. It is characterized by bloody diarrhea with pus and fever. On the other hand, EHEC or enterohemorrhagic E. coli is the opposite, as though it has bloody diarrhea like the EHEC, it has no pus and no fever. Lastly, the EAC or the enteroaggressive E. coli that cause diarrhea in immunocompromised patients. And thus it is non-motile. It was called Friedlander's Bacillus mucus capsulatus because it was first isolated by Friedlander from fetal cases of pneumonia. This is considered as the second most common cause of gram-negative sepsis and pneumonia, which is hospital-acquired pneumonia. There are hospital-acquired pneumonia that is already resistant to antibiotics. This means that they form resistance to the supposed antibiotics that can kill them. And Klebsiella pneumoniae is one of them. Others have been abbreviated as ESCAPE or E for Enterococcus faecium, S for Staphylococcus aureus, K for Klebsiella pneumoniae, A for Acinetobacter baumannii, P for Pseudomonas aeruginosa, and lastly E for Enterobacter species. These including all bacteria belonging to Enterobacteriaceae. The next example is S. typhi. Salmonella typhi differ from other Enterobacteriaceae because it is non-lactose fermenter. Because instead of lactic acid, its fermentation product is hydrogen sulfide. It has more or less similar property with the E. coli as it also affecting the GI, but it can be differentiated from the E. coli as patient having this bacteria has a foul rotten egg-like breath. This is because the fermentation product of Salmonella typhi is hydrogen sulfide, and hydrogen sulfide is a colorless gas that emits a rotten egg-like smell. When the water or food taken was with Salmonella typhi, patients can have a typhoid fever. Signs and symptoms usually include high fever, headache, stomach pain, and diarrhea. Most people who have typhoid fever feel better a few days after they start antibiotic treatments such as chloramphenicol, but was not that effective anymore compared with the other agent. Ciprofloxacin and the newly drug of choice, ceftriaxone, is what is used nowadays. Some rare patients will not be okay upon antibiotic treatment of the three mentioned medicines, so rose spot on the belly can be observed. If this typhoid fever won't be treated on the third or fourth, rose spot on the belly are named as such because they look like small petals of rose, which is small, slightly reddish raised pimples, as shown in the picture. So if the patient are suspected having Salmonella typhi, Windel test can be used to confirm it. The Windel test measures the titer of the antibody against the bacteria which cause enteric fevers such as typhoid or paratyphoid fever. So how they are performing this test? First is the isolated serum from a patient will be tested for H and O antigen titer by placing it on a Dayer's tube for H antigen and Felix tube for O antigen. Agglutination or the formation of mass will means a positive result of infected enteric fever causing bacteria such as typhi. The next example is Shigella dysenteriae. Shigella is similar with Klebsiella that are non-motile because of the absence of flagella. These bacteria are non-lactose fermenter also, similarly with Salmonella typhi because of the enzyme they use which is lysine decarboxylase. Unlike the Salmonella that can be found on cold-blooded animals, Shigella is highly adapted to humans and can only be found on humans and not on the other animals. It doesn't have any other reservoir but its transmission is human to human. We can closely associate this with E. coli. To review again, what is the strain of E. coli which is like shigellosis? Ano na? Sagot? So it's EIC or EIEC. Shigellosis is a severe form of diarrhea and the main site of disease is the large intestine. It has a diarrhea that will result to more than 20 bowel movements within a day. Additional symptoms of infection are abdominal cramps and fever. As similarly with E. coli, its treatment is similar also, which is cotrimoxazole and quinolones. The next example is Proteus mirabilis. Because of its very abundant flagella towards its body, as shown in the picture, it is very motile. This bacterium can swarm or show rapid wavelength movement across a solid culture medium. Proteus colonies bacteria growing on agar exhibit a swarming type of growth, just like in this picture. Swarmer cells with many flagella like Proteus move outward on the edge of the colony and then revert to normal cells with only a few flagella and reduced motility. Periodically, new generations of highly motile swarmer cells develop and the process is repeated. As a result, a Proteus colony has the distinctive appearance of a series of concentric rings. This genus of bacteria is implicated in many infections of the urinary tract and in wounds. Serratia will produce a blood red pigment when grown on agar. It causes endocarditis. Endocarditis is a life-threatening inflammation of the inner lining of the heart chamber and valves. This lining is called the endocardium. Endocarditis is usually caused by an infection. Bacteria, fungi, or other germs get into the bloodstream and attach to damaged area in the heart. Things that make you more likely to get endocarditis are artificial heart valves, damaged heart valves, or other heart defects. Without quick treatment, endocarditis can damage or destroy the heart valves. Treatment for endocarditis include medications and surgery. Initial antibiotic treatment for critically ill patients before cultures and sensitivities for Serratia are ampicillin, gentamicin plus oxacillin, or if the patient is allergic to penicillins, vancomycin plus gentamicin is the initial antibiotic therapy. Let's now proceed to the next group belonging to Prokaryotes gram-negative, which is Vibrionaceae. Like the previous group, microorganisms from this group also can live without oxygen or with oxygen. Because they are facultative anaerobe, what enzyme do we have for facultative aerobe again? That is the supercat or superoxidase and catalase. Since they are motile, we are expecting them to have a flagella to move from one place with the other. But Enterobacteriaceae have peritrichous flagella. In this group of bacteria, they only have one flagellum on their body. Let's review first how flagella is located in the body to have a better differentiation of Enterobacteriaceae with Vibrionaceae. The location of flagella have four types. First is monotrichous. From the word mono means one, these bacteria will have only one flagellum in their body. Lupotrichous are bacteria that have clustered flagella at one end only. Amphitrichous are those bacteria that has either cluster like Lupotrichous or single like Monotrichous, but instead of one end only, it has on both ends. Lastly is Peritrichous. Peritrichous has flagella surrounded its body just like the previous group we have discussed, which is Enterobacteriaceae. So, Vibrionaceae could either be monotrichous or lupotrichous or amphitrichous. Since this bacteria is halophiles, they can live only on the presence of alkaline or saline environment. Usually, this group of bacteria can be obtained on eating seafoods as the environment of seafoods or sea creatures are salty. So there is a high percentage of alkaline or saline environment where they are living. Let's now dive into the different bacteria in this group. First is the Vibrio cholerae and Vibrio parahemolyticus. These two bacteria have a monotrichous flagellum. Since this is monotrichous flagellum, we are expecting that they only have one flagella at their end. Vibrio cholerae is the causative agent of cholera and considered as non-invasive infection. So these bacteria are believed to not spread to internal organ and other part of the body as they are non-invasive infection. Non-invasive infection means they cannot spread out. By triggering the enzyme adenylyl cyclase, which result in the increase of secretion of chloride. As a golden rule, when there is salt, water follows. So since the amount of chloride inside the intestine, the water will also be released in the intestine resulting in watery diarrhea. Vibrio parahemolyticus can be obtained by eating contaminated raw fish, like the sushi. This is the reason why it is advisable to always cook the seafood you eat, as some bacteria like Vibrio naceae bacteria are still present in the body of fishes. The next example is H. pylori. H. pylori is the most common cause of peptic ulcer in humans and can also cause stomach ulcer. How is it affecting its host? The Lupotrichous flagella of H. pylori allows them to colonize on the gastric mucosa or the membrane of the cavity part of the body, such as on the gastric mucosa. And it covers the cavity part of the stomach. Once these bacteria are already on the gastric mucosa, it neutralizes stomach acid. We know that the stomach is very acidic, right? It has the hydrochloric acid that is acidic enough to kill some bacteria. But H. pylori is different. It releases ureas, making the stomach neutral, thereby allowing the bacterium to grow in the stomach. Our body always has its homeostasis. So the time that H. pylori made the stomach neutral, the stomach homeostasis tendency is to secrete more acid. And so, it releases more acid resulting in hyperacidity that result in gastritis and ulcer. Ulcers are like wounds in the stomach that leave hole in its lining. So this is basically because of the too much acidic inside in our stomach, leaving wound in the stomach, and thus it's called ulcer. The last example in this group is Campylobacter jejuni. Campylobacter jejuni has an amphitrichous flagella. It can be acquired by eating uncooked poultry and unpasteurized milk. Since we know now that eating food uncooked may have still bacteria inside, like the Campylobacter jejuni for uncooked poultry and milk, or the Vibrio hemolyticus for uncooked fish, what lesson could we learn from it? We need to properly cook all our food intake, especially the fishes or the seafoods and poultry. The treatment for C. jejuni is quinolones. Let's now proceed to the next prokaryotes gram-negative, which is zoonotic. From the word zoo or zoo, we know that these microorganisms are from animals. These bacteria are infectious diseases that is from non-human animal that is transmitted to humans. The first bacteria belonging to this group is Brucella. Depending on where it comes from, their name also link into. Let's now proceed to the next prokaryote gram-negative, which is zoonotic. From the word zoo or zoo, we know that these bacteria came from animals. These bacteria are infectious diseases that is from non-human animal that is spread out to human. The first bacteria belonging to this group is Brucella. Depending on where it came from, their name also linked into. Like, if it came from goats or sheep, which is the most common type of Brucella, we call it as B. melitensis. If it came from cattle, it is B. abortus. If pig, we call it Brucella sus. If dog, we call it Brucella canis. And all of these types can be treated with doxycycline, streptomycin, and rifampicin. You have to take note that brucella is an airborne disease. This means that once the brucella was on the air, it can go inside the body through an open wound in the skin or through ingestion and inhalation. The next example is Yersinia, which is the Yersinia pestis and Yersinia enterolotica. Pestis causes bubonic plague and pneumonic plague. The term plague is used to describe any quickly spreading fatal disease. Epidemics of all kinds have been described as plague, but the bubonic plague is a very specific disease that first spread around the world in the year 1300s. Bubonic plague is a highly infectious disease spread by fleas that bite their host, usually rodents like rats or in humans, and introduce the bacteria that causes the disease into their host body. Infectious diseases like the bubonic plague that spread rapidly among a community or region within a short period of time are called epidemics. The Black Death is the name given to the first wave of the plague that swept across the Europe in the year 1300s. It is called a pandemic because it spread across many countries and affected many populations. Plague pandemics hit the world in three waves from 1300s to 1900s and killed millions of people. The first wave was called again Black Death in Europe from the year 1347 to year 1351. The second wave is in the year 1500s saw the emergence of a new virulent strain of the disease. The last pandemic at the end of 1800s spread across Asia and at last gave scientific medicine the opportunity to identify the cause of the disease and its means of transmission. Pestis treatment is streptomycin, while treatment for enterolotica is ORS or ORS is oral rehydrating salts, fluoroquinolones, and cotrimoxazole. Another example of zoonotic type of gram-negative prokaryotes is tularensis. Tularemia has more or less similar transmission with Yersinia pestis, only that its most common reservoir is rabbit. But it's not only limited to rabbit, as squirrel can also be its reservoir. The treatment are streptomycin and doxycycline. Next example is Bartonella. Bartonella henselae causes cat scratch disease. Well, because its reservoir is cat. People who own or are close exposed to cats are at risk in having this bacteria. This bacteria was actually inhabit in the red blood cell of the cat. The primary mode of transmission is by scratch of a cat. It is uncertain whether bites of cats or of cat fleas transmit the disease to humans. But the presence of cat fleas is definitely a requirement for the infection to be maintained among cats. Henselae multiplies in the digestive system of the cat flea and survive for several days in flea feces. Cat claws then become contaminated from flea feces. The initial sign is a papule at the infection site, which appear 3 to 10 days after exposure. Swelling of the lymph nodes and usually malaise and fever follow in a couple of weeks. Cat scratch disease is ordinarily self-limiting, with a duration of a few weeks, but in severe cases, antibiotic therapy may be effective, such as azithromycin. Bacilliforme causes Carison's disease, and Quintana causes trench fever. Let's now proceed to the next group, which is non-free living. Why they group as non-free living? It is because they are considered like virus that can only live in the presence of a host. Even other bacteria from this group are thought before as virus because of this property. The first example is Rickettsia. Rickettsia was subdivided by spot fever group, or those patients affected that produce reddish rash, and typhus fever group that produces dark reddish or lesser vibrant than the rashes of scarlet fever. Note also that typhoid fever and typhus fever are two different diseases. Typhoid fever are caused by bacteria from contaminated food or water, while typhus fever is from a vector. Rashes are more common to typhus, while diarrhea are more common to typhoid. Next is Coxiella burnetii. This bacteria causes Q fever. In Australia during the mid-1930s, a previously unreported flu-like pneumonia made an appearance. In the absence of an obvious cause, the affliction was labeled Q fever, or Q as query, because they don't still know what is this bacteria. Much as one might say X fever. So aside from Q fever, some also name this as X fever. The causative agent was subsequently identified as the obligately parasitic intracellular bacterium Coxiella burnetii after several experiments. Next is the chlamydia. Chlamydia are called energy parasites as they cannot make their own ATP. They need the ATP produced by their host to live and get energy from them. They have a unique cell wall. As though they have lipopolysaccharide on their membrane, they don't have peptidoglycan. Haemophilus can be transmitted via respiratory droplets or direct contact with contaminated secretions. These organisms inhabit the mucous membrane of the upper respiratory tract, mouth, vagina, and intestinal tract. The best known species that affect humans is Haemophilus influenzae. Named long ago because of the erroneous belief that it was responsible for influenza. The name Haemophilus is derived from the bacteria required for blood in their culture media. Hemo means blood. They are unable to synthesize important parts of the cytochrome system needed for respiration, and they obtain these substances from the heme fraction, known as the X factor of the blood hemoglobin. Haemophilus influenza is responsible for several important diseases. It has been a common cause of meningitis in young children and is a frequent cause of earaches. Other clinical conditions caused by H. influenzae is including epiglottitis, a life-threatening condition in which the such lesions are an important factor in the sexual transmission of HIV, especially in Africa. Lesions might also occur on such diverse area as the tongue and lips. Next is the Bordetella pertussis. Infection by this bacteria Bordetella pertussis result in pertussis, also known as the whooping cough. The name pertussis is derived from the Latin per, meaning thoroughly, and tussis, meaning cough. In many people, it's marked by a severe hacking cough followed by a high-pitched intake of breath that sounds like whoop. The main target of this bacteria is children. The violence of the coughing in small children can actually result in the broken ribs. Gasping for air between coughs causes a whooping sound, hence the informal name of the disease. Coughing episode occurs several times a day for 1 to 6 weeks. Next is the Legionella pneumophila. Legionella pneumophila usually grows inside the host. Legionellosis disease is characterized by a high fever of 40.5 degrees Celsius, including also cough and general symptoms of pneumonia. No person-to-person transmission seems to be involved in this disease. Recent studies have shown that the bacterium can be readily isolated from the natural waters. In addition, the microbes can grow in the water of air conditioning cooling towers, perhaps indicating that some epidemics in hotels, urban business district, and hospital were caused by airborne transmission. Recent outbreaks have been traced to whirlpool spas, humidifiers, shower, decorative fountains, and even putting soil. So even our air conditioning system or apparatus, so even our air conditioner actually have bacteria, which is the Legionella pneumophila. Let's now go to the thick-walled prokaryotes bacteria. Under here are spirochetes, or their form are spiral-like. Let's now go to the thick-walled prokaryotes bacteria. Under here are spirochetes, or their form are spiral-like. Spirochete from Greek coiled hair, so it's like a curly kinky hair. So for me not to forget that the microorganism under this group, so even our air conditioner actually have bacteria, which is, let's now go to the thick-walled prokaryotes bacteria. Under here are spirochetes, or their form are spiral-like. Spirochete from Greek coiled hair, so it's like a curly kinky hair. So for me not to forget that the microorganism under this group, I always associate them as thick-walled with hamburger or thick hamburger that has bacon, lettuce, and tomato. Or bacon as Borrelia causing Lyme disease, lettuce or Leptospira causing leptospirosis, and tomato for Treponema causing syphilis. So first is Treponema pallidum, or the tomato in our hamburger. T. pallidum is a causative agent of syphilis and has two modes of transmission. Acquired syphilis for sexually transmitted or through the contact in blood, while congenital syphilis is transmitted by mother to fetus. So when the pregnant mother has syphilis, there is a high chance that the fetus will contact the bacteria and may have also syphilis. It is a common pattern of presentation for congenital syphilis or the syphilis on the children, and consists of three phenomena. That's why it's called triad. That is interstitial keratitis, or the inflammation of the middle layer of the cornea, malformed teeth as shown in the picture, and 8 nerve deafness. There may also be a deformities on the nose known as the saddle nose deformity. Again, The Hutchinson's triad phenomenon are interstitial keratitis, malformed teeth, and 8 nerve deafness. Next is Borrelia. Borrelia has 3 strains. Recurrentis are from the word recurring or fever that keep coming back. Burgdorferi causing Lyme disease, which is transmitted through the bite of black-legged tick or deer tick. So once you are infected or been bitten by this tick, the affected area will have a nodule that looks like a mosquito bite when you scratch it that result in a large circular reddish nodule, but much bigger than that of mosquito. Next strain is the Vincenti, which causes Vincent's angina. Next is Leptospira interrogans or the lettuce in our hamburger. This bacteria causes leptospirosis that came from the feces and urine of rodents like mice. We can usually see this on the area with flooded water, and so because there is water and they can live freely in the water area, the feces and urine that has this bacteria will be manifested in that flood. And when a patient with open wound contacted the water on the flood, they will have leptospirosis. The next group is wall-less bacteria. As they have no wall, staining technique won't be possible. So example of these are Mycoplasma pneumonia. Mycoplasma pneumonia is the smallest known free-living self-replicating prokaryotic cell. It produces walking pneumonia or atypical pneumonia. It is also resistant to penicillin, cephalosporin, and vancomycin, and thus its drug of choice is tetracycline. But why is it called as walking pneumonia? Kasi naglalakad yung bacteria? Hindi, joke lang. Walking pneumonia because it is an informal term for pneumonia that doesn't require hospitalization or bed rest. Thus, since patient with the bacteria, though having pneumonia, can just freely walk around. Next group is the cocci. Cocci are a spherical or round-shaped bacteria. Example bacteria for this group is Neisseria. Under this group is Neisseria meningitidis that causes meningitis, and gonorrhea cause gonorrhea. Easy, right? Because of the similar sounding strain and causative agent. So again, Neisseria meningitidis causing meningitis, Neisseria gonorrhoeae causing gonorrhea using an acid alcohol. Example of this is your Mycoplasma tuberculosis. Mycoplasma tuberculosis is the causative agent of tuberculosis. It normally be found on the lungs and can be diagnosed by sputum smear. The treatment for this bacteria is RIPE, or the rifampicin, isoniazid, pyrazinamide, ethambutol, and streptomycin. The treatment for this bacteria RIPE usually lasts for about 6 months, but depending on the case, it may be longer. So the initial treatment of TB is an extensive stage, as in this stage, extensive therapy is needed to kill the bacteria because bacteria can infect also with other humans. The initial treatment will last for 2 months with isolation, and the patient need to take the RIPEs. After 2 months, the patient will no longer have the live bacteria that can infect other humans, and thus after 2 months, there is no need for isolation. But treatment should not stop after 2 months, as there are still other bacteria that live within the infected human. So additional 4 months that sometimes last up to 7 months therapy is necessary. But this time, instead of the RIPE therapy, only the rifampicin and isoniazid should be continued within 4 to 7 months therapy. The last example for this group is Mycoplasma leprae. The last example for this group is Mycoplasma leprae. This bacteria causes leprosy and its treatment are dapsone, rifampicin, and clofazimine. On the next video, we will now continue with the prokaryotes gram-positive. That live within the infected human. So additional 4 months that sometimes last up to 7 months therapy is necessary. But this time, instead of the ripe therapy, only the rifampicin and isoniazid should be continued within 4 to 7 months therapy. The last example for this group is Mycoplasma leprae. This bacteria causes leprosy and its treatment are dapsone, rifampicin, and clofazimine. On the next video, we will now continue with the prokaryotes gram-positive.